Hi, my name is Aisha, and for my biology role model project, I chose Nina Tandon. Nina Tandon was born into a nuclear family of South Asian descent in New York City. She got her early influences in science from her family, with both of her grandparents and most of her siblings being engineers. In school, she studied electrical engineering and then later went to MIT to pursue her PhD. After that, later on she went to Columbia to get her MBA because she wanted to explore the ethics of the biomedical field. She first got into healthcare when she was working on the Human Genome Project, which works on sequencing every gene in the human body. She was inspired to join because of her brother's genetic disease. Some challenges she faced in, in the science field had to do with her cultural background. She reported South Asians having a 1D view of the medical field with only one linear path to success. Another challenge she faced was being a woman in the science field. She had to fight through lots of barriers to get to the place she is today because of her gender. Some hobbies outside of science include musical theater, sewing, and a variety of sports including yoga, rock climbing, and surfing. Nina Tandon credits her creative background into her wanting to work in biotechnology, more specifically cartilage and tissue engineering. She started up the EpiBone company, which works on bone reconstruction by growing cartilage outside of the body to implant it back into a human. Specifically, she works with stem cells. What she does, or what her company does, is take stem cells out of a patient and x-rays the bones that she's needing to replace. They then make a mold or a scaffolding and use the stem cells that they took out to regrow the bone cells. To regrow these bone cells, a specific bioreactor per person is created to mimic the conditions to successfully grow the bones and get them ready to be implanted back into that person's body. Her interest in stem cells led to a publication called Alternative Direct Stem Cell Derivatives Defined by Stem Cell Location and Graded WNTE Signaling, which she published with a couple other people in 2017. This study was created to study how ovarian follicle stem cells, which will be referred to as FSCs, are organized on germanium, which is a part of the ovary in fruit flies. To do this, she studied follicle cells, which will be referred to as FCs, and somatic escort cells, which will be referred to as ECs, which both serve specific functions in the ovaries. To do this, they use multicolor lineage analysis, which is a fluorescent protein to track the lineage of cells inside of an organism. In this photo, they found that the populations of FSCs were asymmetrical. You can see in many of the pictures above, all the different colors were representing different types of cells that were formed from the stem cells. And over the course, the different numbers of each of them and how they changed over about 30 days. They also found that FCs were multiplying quickly while ECs decreased. They found that FCs derive in the posterior or back of the germanium, while ECs derive from the anterior. They found that FSCs move around a lot, and then that the level of WNT, which is a cell signaling protein, dictates the position of the FSCs. Altogether, they found that the FSCs are very unpredictable because they can produce both FCs and ECs and live or die at random. The reason they studied this was because of its similarity to mammalian intestinal stem cells. I chose Nina Tandon as an inspiration to me because she broke barriers to get to the places she's at today. Growing up, there were standards set for her for telling her what she could and could not do. I also loved that she had a bunch of other passions outside of science that led her to become successful in her business. Her work to become a successful businesswoman and scientist resonates with me because I want to do the same. She plans to continue working and expanding her company as well as inspiring other scientists through her TED Talks. Thank you for watching.